gracious. I didn't start recording, but that's okay. We'll start recording now. Okay? So, functions of muscle. All right, now just to let you know, when you think of muscle, think movement. Now, the majority of this chapter, of chapter 10, we will be spending talking about skeletal muscle. Okay, that's going to be the majority. Because cardiac muscle you'll cover when you do the cardiac system, all right, in chapter 19 and 211. Okay, same thing with smooth muscle. Smooth muscle is scattered throughout, so you'll talk about smooth muscle when you cover different systems of the body. Okay, so primarily this chapter, we're going to be spending a lot of time talking about skeletal muscle. So when you think of muscle, think of movement. Now, skeletal muscle, all right, is going to be the muscles that are going to move your skeleton, all right, and they attach, all right, mainly to bones. Okay, but sometimes muscles can attach to skin, and sometimes they can attach to other muscles. All right, so when we think of skeletal muscles, we definitely think of, all right, we're going to move the bones, like for walking or drinking, taking a sip of water or things like that. But also, when we started doing our lab section, we learned that some muscles are used for facial expressions. All right, and when you get into the respiratory system in chapter 23, you're going to learn how certain muscles make it possible for us to speak. All right, obviously breathing, we all know that the breathing muscle, the main breathing muscle, is the diaphragm, okay, but also the external intercostals, okay, are going to also help assist in breathing, okay? So right now, you all are, well, me too, I guess I could say, we're all utilizing the second function up there, maintenance of posture. Okay, helps us, a lot of our muscles, you don't even think about it, help us to stay upright when you're sitting and when you're standing. Okay, when you're laying down, that's usually the time that you're not really engaging your muscles too much. And that's what I tell a lot of patients if they've injured themselves and it's muscle related and it's primarily like a muscle sprain, they pull a muscle in their neck or back. I tell them lay down, don't sit in the chair because you still have to use your muscles. Lay down. Okay, protection and support. All right. Think about this for protection. Now, granted, our thoracic organs are protected by our ribs and the spine in the back and the sternum in the front. All right. But what about some of the organs that are in our abdomen? Okay. There's no ribs down there. Well, part of the protection, all right, for those organs comes from the rectus abdominis, external, internal obliques, transversus abdominis. They offer, all right, some protection to some of those organs there. Okay, and it helps to hold some things in place, so we're going to offer that support portion. All right, now this part here, the regulating and elimination of materials, that's going to be more of a smooth muscle, but there is skeletal muscle involvement. So for example, all right, when you're having to go to the bathroom, all right, for a bowel movement, you have your external anal sphincter is skeletal muscle. You have voluntary control over that. You can, for the most part, can determine when and hopefully where you can use the bathroom, especially if you have to go pretty badly, all right? But then eventually, you know, nature wins out. Hopefully you can find a place. But that's the whole purpose of potty training, okay? You learn to control those skeletal muscles. But when we move things through your digestive tract, that's mainly going to be smooth muscle. Smooth muscle is going to handle a lot of that, okay? Heat production, we talked about that, shivering, all right, for the most part. And when you're exercising, all right, and those muscles are undergoing movements, and in most cases rapid movements, they'll start to produce heat, okay? Mostly unwanted heat, but that's okay because you have perspiration. Sweating helps to get rid of some of that heat, okay? But in certain situations, when you're in a cold situation, shivering, all right, will help to increase your body temperature. So the 5 major functions of skeletal muscle: body movement, maintenance of posture, protection and support, storage and movement of material, and heat production. All right, but the big one is first. Think movement. Muscle begins with an M. Movement begins with an M. It's going to move things. All right. So we talked about movement. We talked about function. Now let's talk about some of the characteristics of muscle tissue, okay? So some of these characteristics you've seen before because we saw them when we were talking about the nervous system, okay? So which is good. Like I said, that's why we do the nervous system first. It makes the muscle system easier to learn. All right, the first one here is excitability, okay? So similar to what we saw in the nervous system, we're going to respond to some sort of stimulus, right? And we're going to change, here's a term that you're all familiar with, the electrical membrane potential. So muscle has a membrane potential also, okay? The value is different, okay? In the nervous system, the value for the resting membrane potential is -70, okay? And in the muscles, we're going to learn, it's a little bit different, but the same type of concept, okay? So how we create that electrical membrane potential is going to be the same. So we'll talk a little bit about that. Conductivity. All right, we know that a neuron can conduct its action potential down the axon. We're going to see how muscle can also send its version of the action potential, all right, along its cell membrane. Okay, and that's how we're going to be able to stimulate the muscle. Right? Now, a neuron doesn't have this next characteristic. It can't contract. It can't change its shape. But a muscle can. So this one here, we're going to be spending a lot of time on, not today, in a later lecture period, all right, is contractility. All right? This is where we're going to learn about the power stroke, the sliding filament theory, how we'll move two different protein, all right, contractile proteins, protein filaments past each other to actually cause a muscle to shorten and contract, okay? So we're going to learn about that, and that contractility is going to result in movement, okay? So when these fibers are moving past each other and creating that contraction, that shortening of the muscle, then we're going to see, as a result, some movement out of that, okay? Extensibility is the opposite. Now we're going to stretch the muscle cell. All right, so we're going to lengthen it. Okay, what goes up must come down, but you know when you're stretching that your muscles have some sort of ability to, I won't call it so much an elastic form, but when you're stretching it, all right, you are lengthening the muscle fibers, all right, to reduce injury. All right, then we have elasticity. So if you contract a muscle, it's going to go back to its original shape, or length, I should say. If you stretch a muscle, it'll go back to its original length. And this characteristic is its ability to return to the original length, okay, following a contraction or if you're stretching. Okay, so there's a couple new characteristics of muscle tissue here, all right, and there's a couple that you've seen, all right, when we looked at the nervous system. Okay, so when we were talking about the nervous system, we said that the neuron was the cell. Okay, the neuron was the cell of nervous tissue. An actual nerve is the organ. Okay, so like the sciatic nerve, for example, is considered to be an organ. Okay, well, in this chapter, our organ tissue is going to be the skeletal muscle. Okay, remember, a definition of an organ is going to be, all right, a structure that is going to be made up of several different tissue types that are going to perform, all right, some functions. Well, if you look up here, you can see, all right, we got muscle, where is it? Muscle fibers, connective tissue, blood vessels, and nerves. Those are different tissue types. And we throw them all together. Now we have our organ, our skeletal muscle organ. Okay? So we're going to start off. This one I have to put my glasses on. All right, I'm going to show you this picture here, and I'm going to kind of work backwards because this part is a little bit complicated. I want to start small and go big. Always start small and go big. All right, so when we're looking at the organization of how we organize the skeletal muscle, we're going to start with the cell first. Whoops. Okay. So we're going to start off, all right, with our cells first. All right, so our cells are going to be our muscle fibers. Okay. So our muscle fibers are the smallest structure. Now what we're going to do is we're going to bundle a bunch of our muscle fibers together. And there's a couple different names here, all right, for the muscle cells. We also will call those myofibers. Okay? So we bundle up. This is an individual skeletal muscle cell, which we saw before, remember in chapter 5. All right, it's striated and whatnot. And you see there's a bunch of smaller structures in there. We'll get to those in a bit. Okay, so the muscle fibers we bundle up. All right, we have a bundle of them, and we call those fascicles. And those are just bundled muscle fibers. Then we take several muscle fascicles and we bundle them up, and we get our overall skeletal muscle. Okay, so the whole muscle is going to be made up of several different muscle fascicles. Okay, and a muscle fascicle is going to be made up of several muscle fibers. All right, or myofibers. That's the skeletal muscle cell. Okay, so this should make that a little bit easier to understand here what I'm talking about. Okay, let's start with the smallest one. All right, our muscle fiber, make sure that you know that, is a muscle cell. Okay, we also call them myofibers. All right, now there's smaller structures inside. We'll get there. I don't want to throw too much at you. Okay. So then when we bundle up a bunch of the muscle cells or the muscle fibers, then we have a fascicle. And then when we bundle up a bunch of fascicles together, that gives us our whole muscle. Okay, so there's a hierarchy here. An increase in the level of organization. Okay, so let's not forget our rule of 3. So remember when we were talking about the neurons and then our nerve bundles and then the nerve all together? There was 3 layers of connective tissue, okay? And actually, most likely you'll see one of those on the test, or for the lab test, I should say. All right, the endoneurium. Remember, the endoneurium covers each individual neuron, helps to insulate it, okay, and separate it from other neurons. Okay, so we're going to see something similar to that here with the muscle. All right, we have 3 layers. Okay, their job is to protect the tissue inside of them. All right, so I'm going to go start down here with the smallest and then work my way up. So the Endomysium, this is that connective tissue layer that's going to wrap around each individual muscle fiber or cell. It's a loose connective tissue. Okay? Well, guess what? Endoneurium was loose connective tissue that individually wrapped around each neuron. Same thing. Okay? So, and it's going to offer similar type of function. It helps to insulate, all right, the muscle fiber, much like the endoneurium. Okay? We're also going to have some blood vessels inside there, which is great because we have to have a way of nourishing our tissue in there. All right? And it helps to give attachment sites for other muscle fibers that are close by. Okay? The next layer now, remember what I said, we're going to take a bunch of muscle fibers, muscle cells, and we're going to bundle them up, all right, and create a fascicle. So the covering that goes around the fascicles is perimyosim, and it's made up of dense irregular connective tissue. Well, guess what? So is the perineurium, dense irregular connective tissue. Same thing, okay? So here we're going to see blood vessels and nerves. And then finally, we're going to wrap the whole muscle up in the epimyosim, and that's again another dense irregular connective tissue, and it's going to cover the whole muscle. Okay? And the epineurium did the same thing. Same type of connective tissue. What I mean by that is dense irregular connective tissue. All right, for the epineurium and for the perineurium. Same thing here. All right, we have dense irregular connective tissue covering the epimyosin and the perimyosin. Right, and then for the endoneurium, inside the nerve, all right, that was made up of a loose connective tissue, the areolar connective tissue. We see the same thing here. In our endomysium, we're dealing with a loose connective tissue. Okay? So here again, you can see, all right, this is showing you, all right, the coverings here. This is that individual muscle cell. It's got the endomysium covering it, that loose connective tissue. And you can see here's our fascicle. And it's covered by the perimyosim, and that's going to be the dense irregular connective tissue. And then finally, the muscle as a whole is going to be covered by the epimyosin, which is another dense irregular type of connective tissue. All right, let's talk about some associated tissues, all right, when we are dealing with our skeletal muscle organ. Okay? So, well, it's up here on the board. It doesn't help me to say that. But tendons attach muscles to bone. Ligaments attach bone to bone. Okay? So, a tendon is going to be a cord-like structure. And we know that it's made up of not dense irregular, but dense regular. Ligaments are made up of the same type of connective tissue. Okay? So again, tendons attach muscles to bone, muscles to other muscles, or muscles to the skin. Okay? And tendons are made up of those three connective tissue layers that we just saw. So the tendons, all right, what we'll see is the epimyosin, the perimyosim, and the endomysium, they'll all converge, all right, and they will help to form the tendon. Okay, they all come together. Now, we already saw what an epineurosis is. It's just a flat version of the tendon. Same thing. It's got the three connective tissue layers also. All right, so a couple other new names that we haven't seen before, the deep fascia and our superficial fascia. Okay, the deep fascia, all right, is going to be, all right, the connective tissue layer that covers the epimyosin. Again, it's made up of another dense, irregular connective tissue. So if you were to be dissecting a cadaver, all right, and you pull everything away and you get down to the actual muscle, okay, and you can actually see the muscle. Well, there's this layer of connective tissue over it. It's called the deep fascia. It helps to separate, all right, muscles in the same vicinity. For example, when we were just learning about the muscles of the anterior or flexor compartment in the forearm on Tuesday, all right, the deep fascia is going to separate, okay, the pronator teres from the palmaris longus, or it'll separate the palmaris longus from the flexor carpi radialis. Okay, so it helps to separate these muscles, all right, from each other. Okay, especially when they're in the same compartment. All right, superficial fascia is going to be, all right, this layer of connective tissue, right, that is going to be just on top of the deep fascia. Check this out. Look at that. Areolar and adipose connective tissue. Does anybody remember seeing that before? Give you a hint. It's in chapter 6, which is the skin. Hypodermis. The hypodermis, the subcutaneous layer. Subcutaneous layer, also known as the superficial fascia. We used to think it was junk. Anatomists, when they were dissecting cadavers, they just threw it out. Superficial fascia was junk. All right? But it's not. Okay? So this layer here literally is going to separate your skeletal muscles from the skin. It's superficial to the skeletal muscles, deep to your skin. Okay? Areolar and adipose tissue, both loose forms of connective tissue. All right, and then finally, the other tissues that we're going to find in our skeletal muscle organ are going to be blood vessels and nerves. Okay? So keep in mind, skeletal muscle tissue is highly vascularized. We have an extensive amount of blood vessels located here, which only makes sense, okay? Because those muscles, especially if they're undergoing a high metabolic rate, they need to get plenty of nutrients. And at the same time, they need to get rid of the waste products that they're producing. All right? Skeletal muscle is also going to be innervated by, you need to know this, somatic neurons. Somatic neurons are the neurons that you can consciously or voluntarily control. It's part of the somatic nervous system. All right? So that gives us voluntary control over these muscles. All right? And so these neurons are going to end right at where the actual synaptic knob is going to synapse, right, with the muscle itself, the muscle cell itself, I should say. And we refer to that as the neuromuscular junction. We'll talk more about that when we talk about how we stimulate skeletal muscles. Okay? So we'll see, and you'll see it. Well, I don't know if I have a picture of it here. Of course I have a picture of it here. It's going to look well, I can't believe I don't have a picture of it. Boo! All right, I don't have a picture of it. Okay, no worries. All right, so question for you. What are the locations of the endomysium, perimyosim, and the epimyosim? All right, endo, all right, is going to be on the outside, so it's going to cover the whole skeletal muscle. All right, and it's going to be made up of that dense, irregular connective tissue. The middle layer, the perimyosin, okay, is going to be wrapping around the muscle fascicles. But in those muscle fascicles, we'll also see bundled up in there into each fascicle blood vessels and nerves. And then the inner layer, the endo, all right, is going to be surrounding each individual muscle fiber, which is the cell. And, yeah, you're right. It should be epi. Yep, thank you. Epi. You're the first person that's pointed that out. I just usually go along on that. Yeah, the epimyosin. Okay. Oh, excuse me. That is, hold on, I marked out the wrong one. This one should be epi. Epi's on the outside, like an EpiPen. All right. The endomysin on the inside is going to individually insulate each of those muscle fibers from each other. Okay. So kind of similar to what we did in chapter 12, we talked about the anatomy and the different parts of a neuron. So I want to do the same here and talk about some of the individual parts of our muscle cell. Okay. Now you know, because remember how we talked about in the axon, we had the axolemma, which is just a specialized name that we gave to the plasma membrane in the axon. Well, we're going to rename our plasma membrane in our muscle cell, and we're going to rename the endoplasmic reticulum, all right, in the muscle cell. Okay, so there's a couple new terms here. You're familiar with some of them, okay? But I'm going to give you a second to write this down. Did I skip? No, I did not. Okay. Okay. So the cytoplasm in a muscle cell gets a special name, and we call that the sarcoplasm. This is where, and this is important that you know this, all right? As we know, in the sarcoplasm, in the cytoplasm of the cell, that's where all the organelles are located. You know, the Golgi apparatus, the peroxisomes, the lysosomes, endoplasmic reticulum, yada, yada. A bunch of cellular organelles are going to be there, right? Well, in addition to the organelles, all right, in a muscle cell, we are going to see, this is where the contractile proteins are. Okay? So we're going to have, when we talk about the cross-bridging and how we're going to create, all right, a muscle to contract, all right, when you hear me talking about the contractile proteins, which are actin and myosin, all right, those proteins are located in the sarcoplasm, which is the cell cytoplasm of a muscle cell. Okay? Also, inside the cell, we'll see our nuclei, but as you remember from chapter 5, these cells are multinucleated. And at one point, all right, when you were developing inside of mommy, okay, you had these individual cells called myoblasts. Okay, they were the embryological stem cell for our skeletal muscle cells. Well, they just started fusing, okay, but none of them wanted to give up their nuclei, so they just fused into one cell, all right, and that is why we have multiple nuclei, because of all these individual myoblasts fused together. We do have another group of cells, and we'll talk briefly about the satellite cells. But the satellite cells are like the support cells. Right? We saw the glial cells for the nervous system help to repair and nourish and remove waste and help to protect. All right? In this type of situation, all right, the satellite cells have a similar role in which they help to support, all right, the skeletal muscle cells, and if they get damaged, they help to repair the skeletal muscle cells. All right, another term for that we're going to learn now for the plasma membrane, and you want to really know it because I'll be mentioning this quite a bit in the future here, is the sarcolemma. The sarcolemma is what we call the plasma membrane, all right, for our skeletal muscle cell. So there's a couple new characteristics for this. In our sarcolemma, we have these structures which we call tubules or transverse tubules. And basically, it's a hole in the plasma membrane that invaginates or pushes down into the cell. Right? And that plays a role in trying to excite the muscle cell. Right? When we're going to try to stimulate it to contract. Okay? Now, along your plasma membrane, or excuse me, the sarcolemma, and along the T-tubules, we have voltage-gated ion channels. All right, so we've seen voltage-gated ion channels extensively in the neurons. Okay, you thought you could get away from them, you can't. They're also here in our skeletal muscle cell, and their job is the same job that you saw, all right, in the neuron. Its job is to conduct the electrical signal, all right, along the plasma membrane or the sarcolemma, much like we saw the action potential getting conducted along, all right, the axolemma of the axon. Okay, so the same type of concept. Okay, voltage-gated ion channels here in our skeletal muscle cell are going to be similar to what we saw in the neuron. Okay. So, and another new term that you're going to see is voltage-sensitive calcium channels. Okay. Now, here's the thing. Calcium plays a huge role in muscle contraction. All right, we learned about its important role in last class in the nervous system because it was our voltage-gated calcium channels that opened up in the synaptic knob that allowed calcium to enter into the synaptic knob and promoted the secretory vesicles with the neurotransmitter to move to the plasma membrane and release the neurotransmitter into the synaptic cleft. Okay, so we saw the importance of calcium for that. We've seen the importance of calcium in building strong bones, okay, because it's part of hydroxyapatite. All right, now we're going to see its importance in actually, all right, causing a muscle contraction. All right, you want to consider, all right, calcium. All right, think of it as the princess. We're going to go fairy tale wise. Think of it as the princess that's trapped in the tower, and we have to rescue the princess. We have to get the princess out of the tower, all right? Or it's Jack Sparrow in jail in the dungeon somewhere, and we have to get. Does everyone know? Does anyone know? All right, Jack Sparrow, Pirates of the Caribbean. We have to release calcium because it's in prison, in a resting muscle, because we can't have a muscle contraction without it. Okay, and we'll talk about what type of role, all right, that calcium plays. All right, but ideally, it's these voltage-sensitive calcium channels that are going to do that. They're going to release or free calcium that's currently being trapped, all right? And the only way to do it is to have an action potential, stimulate the openings to those voltage-sensitive calcium channels, okay? So we need to get our action potential that's come from our neuron and that's going to be traveling along the sarcolemma or the plasma membrane of our skeletal muscle cell, and we need to get it to these voltage-sensitive calcium channels so we can release the calcium that's trapped. That's our goal. There's a lot to get us there. So you should all, by the time we're done with this chapter, you're all going to be blown away at how awesome your skeletal muscle cells are and able to do this. All right, so here's just a quick picture you can see, all right, embryological development, all right, when you're just an embryo and you have all these myoblasts, all right, they will fuse together and eventually form, all right, one single muscle fiber, but they will maintain their individual nuclei. All right, and then you have your satellite cell, your support cell. Okay, it's going to help to repair any damage and support the skeletal muscle fiber. Okay, big picture here. So if you look here, what I am going to show you now is, all right, here's our skeletal muscle. Here are these fascicles. We're going to pull out one fascicle, and now we're going to pull out one individual muscle fiber, which is one muscle cell. So now we're going to zoom in and take a close look at our muscle cell that we just talked about. All right, now you can see there's some other structures in here. I'm going to get to this. We'll talk about the myofibrils here in a second. Okay? All right, so covering our muscle cell is going to be, you can see we peeled it back a little bit. That's the sarcolemma. That's our plasma membrane. Okay, and it surrounds, all right, this is one entire cell. You can see the nucleus here. Then you've got these little tube-like structures, which we'll talk about. Okay, so also you can see these little holes here. All right, those are the openings to the T-tubules. All right, so it's an opening to the T-tubule that goes down inside the cell. Much like your chimney, if you have a house with a chimney on it, the chimney goes down into your house. All right, that's what the T-tubule does. It goes down into the cell. Okay, so on this drawing here, the T-tubules are these green structures right here, or greenish tubes here. And then on either side of those green tubes, you can see a purple tube. All right, we're going to talk about what that is in a second. All right, those are referred to as what we call, I'll talk about it in a brief second, the terminal cisterna. Okay, and think of the terminal cisterna as these reservoirs for calcium. Okay, calcium sits inside of that, inside of these structures. And then if you look, well, I'll zoom in more. Okay, so here are these green tubes. Those are the T-tubules. Okay, and then you can see on either side of them is the terminal cisterna. And then extending from the terminal cisterna is this mesh-like, all right, this tubular network that goes all over the place. Here's another, all right, terminal cisterna on the other side, but there's these little tubes that go back and forth. Okay, and those are referred to as the Sarcoplasmic Reticulum. Similar, remember the endoplasmic reticulum? Smooth endoplasmic reticulum and rough? Similar to that. It's just a tubular network. And there's calcium that are in these tubes. But here's the thing. These tubes, all right, are separate from the sarcoplasm. Okay? They're inside, but they're separate. So the calcium is trapped inside of these tubes. All right, zoom it out here. Okay, so now what we're going to do is we're going to take one of these little tube-like structures here that you see, and we're going to slide it out. And we're going to look at that, okay? And that structure there is a myofibril, okay? Myofiber is a muscle cell, all right? Or we can also refer to it as a muscle fiber, right? And then inside of the muscle fiber or the muscle cell, you have all these little tubes. And so we're going to slide it out, and you can see that this individual tube is a myofibril. Okay? So we look here at our myofibril. We zoom in on it. Okay, we pulled out one of these tubes. Here it is. Here's our myofibril. All right, and you can see here you have these tiny, now we're going to get even smaller, the myofilaments, which are the protein filaments. What are the myofilaments? Those are your contractile proteins. That's actin and myosin. It's these myofilaments and how they interact, which is going to cause the contraction. Okay? So there's this structure here called the sarcomere. That is the contractile unit. All right? It's one single unit, and we'll break it down, its anatomy here. But it extends from, this is called the Z disc or the Z line. It goes from one Z disc to another Z disc. And it's these, all right, structures that when they shorten because of contraction, all right, because of these protein filaments are going to overlap one another. We'll get into that. I'm jumping ahead. Okay, so you have this myofibril, and then on the outside of it, much like my shirt sleeve, and my arm is the myofibril. My shirt sleeve is going to be this purple and green structure that you're seeing here, which is the sarcoplasmic reticulum, which is this tubular network. That's where calcium is, stored in there. Okay? We'll get into more detail here. Okay. So what we're doing here is we're zooming in and showing you, here's the sarcolemma, all right? And then you can see here is the opening to the T-tubule. You can see how the T-tubule goes down and into the cell. But along the sarcolemma, we have voltage-gated ion channels. Well, guess what? We've seen these before, voltage-gated sodium channels and voltage-gated potassium channels. Same role, okay? Meaning they're going to let sodium and potassium move across the plasma membrane, okay? But when we get down here into the T-tubule, we're going to see these voltage-sensitive calcium channels. More on that, okay? But it's these voltage-sensitive calcium channels that are going to release calcium that's being stored in these purple tube structures that we saw on the previous slide, all right, the terminal cisternae. All right, we want to get calcium out of this purple tube structure and into the sarcoplasm. That's our goal, because that's where our contractile filaments are, the contractile proteins. Okay, so a couple more definitions here as we're talking about the muscle cell. The myofibers, all right, are going to be those bundled myofilaments, which are actin and myosin. Okay? And these myofibrils are surrounded or enclosed by the sarcoplasmic reticulum. And that sarcoplasmic reticulum is that tubular network. All right? We saw it in other cells. It was similar to the smooth endoplasmic reticulum there. All right, so this is going to be a horrible drawing. All right. So there's my myofibril, and then I'm going to have all these little tubes going all over the place from end to end. All right, that's the sarcoplasmic reticulum. On either end, though, of the sarcoplasmic reticulum, all right, you're going to have this blind sac. It's referred to as a blind sac, but it's just that sac-like structure. That's this, right here in this drawing, this thing, here. We zoom out a little bit more, it's this. See the green tube here on either side of the green tube. Okay, on either side of this green tube, the green tube is the T-tubule, on either side, that's the terminal cisternae. And then in between these two terminal cisternae, you've got these tubes running across. That's the sarcoplasmic reticulum. Okay? All right. Now, it's the sarcoplasmic reticulum that is going to hold on to, and this includes the terminal cisterna, the calcium ions. They're trapped in there. Okay? We want to get them out. All right? Well, they got put into the terminal cisterna by the calcium ion pumps. The calcium ion pumps, their job is to pump calcium out of the sarcoplasm of the cell and into, all right, the terminal cisterna. And then once they go into the terminal cisterna, all right, they will bind onto these two storage proteins called calmodulin and calsequestrin. I always remember calsequestrin because you've heard of terms when they talk about a jury, they're like they've sequestered the jury, all right, so they keep the jury for whatever reason in a court case. Well, calsequestrin is going to sequester or hold on to calcium in, all right, the terminal cisterna. Okay? All right, so keep that in mind. So this, using this, all right, analogy again, the shirt sleeve, okay? Think of the sarcoplasmic reticulum as a shirt sleeve. Think of the myofibril, all right, as the arm for that shirt sleeve. All right, and then you could think of the terminal cisterna as the hem or the cuff at the end of the shirt sleeve. All right, and finally, we have to keep in mind our calcium release channels, because our goal is to get the calcium out of the sarcoplasmic reticulum and the terminal cisterna when our action potential arrives. So when that action potential, that electrical signal travels down the T-tubule, it's going to stimulate those voltage-sensitive calcium channels to open up, and that will cause these calcium release channels to release calcium into the sarcoplasm. That's our goal. Our whole purpose is to get calcium into the sarcoplasm. Because when that occurs, you need two things for a muscle contraction. You need calcium and you need ATP. If you do not have either one of those, you can't have a muscle contraction. Okay? You need calcium and ATP. All right, so that's what this is talking about here when we're talking about the triad. Those two terminal cisterna and then the T-tubule in the middle. So our action potential will come down here. Just pretend that this is the sarcolemma, the plasma membrane. So our action potential travels down, all right, the sarcolemma, then it comes down into the T-tubule, and it stimulates the opening here of the voltage-sensitive calcium channel. All right, that will cause a shape change to that channel, and it will literally open up our calcium release channel that's located on the terminal cisterna. And then the calcium that's in, all right, the terminal cisterna and the sarcoplasmic reticulum will leave and go into the sarcoplasm. And that's where our contractile proteins are. All right? And then when our contractions, can you go back to the last slide? I didn't get what. Oh, okay. Yeah. Let me just finish this thought real quick. When we're done with our muscle contraction and it's like having a party, you got to clean up after the party, all right? There's calcium that it's out here into the sarcoplasm. We want to get that calcium that's in the sarcoplasm back into the terminal cisterna and the sarcoplasmic reticulum. Then we've got our calcium ion pump here that is going to return it back into the terminal cisterna. Much like you saw the calcium ion pump in the synaptic knob, all right? It was pumping calcium out of the synaptic knob after an action potential because it was trying to create that concentration gradient there. All right, questions about that so far? Probably many, but you probably just don't know what. Keep in mind, our goal is to get calcium into the sarcoplasm because that's where the contractile proteins are. That's where the actin and myosin are. Okay, that's our goal. In a resting muscle, calcium is trapped, and we have to release it. And this is how we do it. I'll go into more details here. All right, so what are those myofilaments? All right, we just talked about what covers the myofilaments, the sarcoplasmic reticulum and the terminal cisterna. So the myofilaments are the contractile proteins made up of actin and myosin. Okay, these are, we briefly talked about this when we were talking about in chapter 4 about the cytoskeleton, okay? And so when we were talking about myofilaments, all right, or the different types of filaments inside of cells, well, this is a group that are specific to, because myo means muscle, to a muscle cell. Okay, so there's two different types of proteins that we're going to deal with. Let's talk about the first one, myosin, which is also called the thick filament. Okay, and I'll show you a quick picture here. Actually, this is a perfect opportunity to show you You kidding me? Okay. Hello, are you there? Okay. I think you guys can hear me now. I think you may have missed that video. All right. All right. So let me just pick up right here, and then we'll continue on. And if not, then I can go over the video in lab or something. All right. So our myofilaments. All right. The goal is to get the calcium into the sarcoplasm where these myofilaments are waiting. All right. So the two types, the thick filaments, which are the myosin. All right. These are the ones that have that globular head. Okay. And it's this head that is going to attach onto the other protein filament, the actin. Okay, so myosin looks like a bunch of just ropes tied together, and then it has, so at the end of this rope, you'll have this globular-like head. Okay, and we just have a bunch of them. Okay, and they all come off of these rope-like structures. Okay, so the rope part is the tail, and then you have the globular head at the end. All right, now on the head, all right, you have a special binding site, all right, for the molecule of ATP. And there's also an enzyme that sits there that is going to break ATP down into ADP, an inorganic phosphate. Okay, so it's that enzyme, and we'll talk more about that, but it's the enzyme that helps. Because remember, when we break down ATP, when we snap off one of the phosphates, that's when we get the energy. Remember, we need energy to do this stuff. Okay, and we're going to talk about that more in detail.
Go away,
yep.
Or do they just go to the next?
Nope, they'll pop off and go into the cytoplasm,
Okay.
and then the cell will recycle it, the mitochondria will recycle it, and they'll turn it back into ATP. And we'll talk about how that does that too. Really cool. There's a couple of different ways. The other filament is the actin, which is the thin filament. Okay? So the actin, imagine taking two pearl necklaces and just twisting them around each other. Okay? One is called F-actin and the other one is the G-actin. Now it's the G-actin that has the binding site for the globular head for myosin. Okay? Now we saw in that video how that myosin will extend across and bind on, and then it kind of pulls things past it. And we'll talk about that. Okay? So just know that the G-actin has the myosin binding site for the myosin head to attach to. Now, in a resting muscle, okay, this binding site is blocked. Okay? It's blocked by this protein in particular, tropomyosin. Attached to the tropomyosin is troponin. So, all right, so I'm going to come right back to the slide here. So here's what we're looking at. Okay, we're going to the thin filament. Okay, this is the actin. See, it looks like two pearl necklaces we twisted together. And the myosin binding site are these dots here. Well, right now in this picture, it's blocked by this rope-like protein. This rope-like protein, that's tropomyosin. Okay, attached to the tropomyosin is the troponin. You see this little cup here? That's where the calcium that we saw in that video, it will come on and sit on there. When it does that, it shifts these ropes off of the myosin binding site. So now the myosin head here can attach onto actin. So that's, my whole point was when I was saying that this is why calcium is important. We need calcium to come and sit on the cup here and move this rope protein, the tropomyosin, off the binding sites. If you don't have calcium, you can't move the tropomyosin off of the myosin binding site, and we can't get a cross bridge. All right, so that's why it is so crucial to have, all right, calcium to do that. Okay, so tropomyosin is the protein that covers the binding site. Troponin has the binding site for calcium. And together, we call them the troponin-tropomyosin complex. Okay? These are referred to as regulatory proteins. So you need to know that tropomyosin and troponin are the regulatory proteins. They regulate this process of muscle contraction. Okay, so as you look here, all these myofilaments are just bundles of these thick and thin filaments in close proximity to one another. Okay, the myosin, which is the thick filament, has these globular heads, and they have binding sites for actin, where we're going to attach to the small protein molecule here. And then also it's got a binding site for ATP. So the ATP is going to help power this thing through. But there's an enzyme, we'll talk about it in a second, there's an enzyme that sits on this binding site, the ATP binding site, that's going to break down the ATP into ADP and an inorganic phosphate. All right, so you heard the term sarcomere. All right, this is, all right, the functional contractile unit of the muscle fiber, all right, of the myofilament, I should be more specific, okay? So they're just basically a repeating unit that we just stack end to end to end. So if I show you a picture here, that's what we're seeing here on this diagram. So we've pulled out, again, our myofibril, all right, from our muscle cell. And basically, we have, here's one sarcomere. Then there's one next to it. Then there's one next to it. We're just stacking them end to end to end. All right, that's what we're talking about. So basically, a myofilament is just made up of a bunch of sarcomeres that are just added on to one another. Okay? So in the sarcomere, we are going to have areas of thick and thin filaments that are going to overlap one another. So basically what makes up the boundaries of a sarcomere is going to be the Z disc of one end of the sarcomere all the way to the Z disc of the other end of the sarcomere. So it'll be that distance. And we're going to talk about what the Z disc is. But the Z disc is where our thin filaments attach themselves to. So if we're looking here, here's a Z disc, also known as a Z line, okay? Z disc, Z disc. All right, here's our thin filament. Here's another thin filament. Here's another thin filament. They attach or anchor themselves to the Z disc. So one sarcomere is this whole distance from Z disc to Z disc. Oops, one more. Okay. So here's the thing. Certain parts of the sarcomere, we were going to see an overlap between the thick and thin filaments. In certain areas, we're going to see just the thin filament. In certain areas, we're going to see just the thick filament. All right? But this is what gives skeletal muscle its striated appearance, that there's alternating areas that are light and alternating areas that are dark. So it gets it that striated appearance. Okay? So now we're going to talk about what constitutes some of these alternating dark and light areas. Okay? We have what we call I bands and A bands. All right? And it all depends on where the thick and thin filaments are located. Are they overlapping with another filament, or is it just the thick filament in one spot, or is it just the thin? So we'll start off with the I bands here. Okay? The I bands are going to be a light appearing region because they only contain the thin filament. Let me come show you here real quick on our picture. Okay? So here's our I band. You can see at the center of the I band is the Z disc. So we just see thin filaments in this area. Okay, so more light can pass through it when we look at it under a microscope. Okay, so when we look at it under the microscope, we're just going to see the thin filaments only. At the center of the I band is the Z disc. Now, when the muscle contracts, because we're going to start to get overlapping of the thick and thin filaments, this area will get smaller and smaller until, all right, it can possibly disappear altogether, all right, during maximal muscle contraction. Okay? The next part is the A band. All right? Now, important that you know this. It does not change length during muscle contraction. Okay, it does not change length. So we'll see thick filaments here, and we'll see the thin filaments overlapping in that area. This will be found in the middle of our sarcomere. So let me show you a picture here of it. Whoops. Okay, here's the A band. Okay, so you can see at the center of the A band, we have our M line that the thick filaments are anchored to. Okay, but in the A band here, we're going to see that's where we're going to have overlapping here of the thick and thin filaments. Okay? All right, that does not change length during our muscle contraction. And because there's overlap, and I should point this out, it will have that dark appearing section on the light microscope. All right, the H zone and the M line. The M line, that's where we attach our thick filaments. It's at the center of the sarcomere. You can see it right here. Here it is, right at the dead center. And then on either side here is the H zone. And the H zone is going to be where we just have thick filaments only. Okay? And again, this is one of those areas that completely goes away, all right, when we have maximal muscle contraction. And that's because, all right, we get overlap in that area because the thin filaments slide past, all right, all the way up to where it almost makes connection with the M line there. Okay, in which band are there thick filaments only with no thin filament overlap? That's going to be the H zone. That's where the M line is the central portion there. All right, a couple other proteins that you need to know that are going to be found here in the sarcomere are connectin and dystrophin. Now, connectin, and it's really cool, I thought it was clever how the book drew this in, all right? It looks like a spring. Okay, so because it looks like a spring, it's going to provide the elasticity to the sarcomere. So if you check it out here on our picture, that's this guy right here. Okay, so it's drawn in like a spring, but you can see it attaches onto the Z disc, and it attaches onto the end of the thick filament. And that protein there allows for that muscle to return back, all right, to its original shape when it's stretched or when it's contracted, okay? And then dystrophin, you probably heard of muscular dystrophy. This is that protein, all right, that if it is abnormally created, it will mess up or cause the organization of the sarcomere to be imperfect or poorly organized, causing abnormal muscle function. And in certain cases, depending on what you have, there's Duchenne's muscular dystrophy, in which you get abnormal deposits of protein in the skeletal muscle. It doesn't mean the muscles look bigger, but they're not stronger because the sarcomeres or that muscle tissue is pretty much useless. But the dystrophin here is where we're going is going to offer an anchoring point, all right, for some of these myofibrils here. And when we have that, then it's going to cause, or when we have irregularities with that, like with muscular dystrophy, we're going to have irregular muscle function here. So that's the dystrophin. And you can kind of see it on this cross section here, all right, throughout the different portions of the sarcomere here. All right. So, for example, you can see it here by the M line. All right, we've got our accessory proteins here, and you can see how it attaches onto some of these thick filaments. But basically what it does is it helps to create organization here throughout the sarcomere. So when you have the the muscle fibers sliding past one another, all right, they're not banging into each other. All right. So I'm going to get more into the detail here about how we produce energy for muscles, especially for muscle contractions and whatnot. But I want to briefly mention it here, and then we'll talk about it after next class, okay? We learned in chapter 4 that the powerhouse of the cell is the mitochondria. And it's the mitochondria that is going to produce the majority of the energy for metabolism, muscular functions, because that is its job, especially during aerobic cellular respiration, is to produce ATP. So when you're thinking of the mitochondria involvement in energy production, you have to think of aerobic cellular respiration. Aerobic means with oxygen. Okay? We need oxygen for this to happen. Without oxygen, then the cell creates energy through anaerobic cellular respiration. And for example, one glucose molecule with aerobic cellular respiration yields 32 ATP. One glucose molecule with an anaerobic cellular respiration yields 2 ATP. So you can see the importance of oxygen here, okay? Well, in our blood, we've got a special protein that carries oxygen. It's called hemoglobin, okay? Well, our cells, not wanting to be left out, have their own version of the oxygen-carrying molecule, all right? But in the muscles, we call it myoglobin. And this protein molecule is going to be used to store oxygen so we can undergo aerobic cellular respiration. So we can produce those 32 ATP molecules, okay? Also, remember there's 2 organs in your body, or 2 tissues in your body that are going to store glycogen. Glycogen is the storage form of glucose. The liver is the primary organ. The other organ is skeletal muscle. So glycogen is stored glucose that's on hand inside the muscle cell. Well, that's great because we can start breaking down the glycogen, right, into glucose, and then we can metabolize the glucose so we can create ATP. This creatine phosphate is another molecule. If you notice, the second half of the name is phosphate, all right? Well, ATP is adenosine triphosphate. So basically, this molecule will gladly and quickly give up its phosphate molecule and donate it to an ADP molecule to make more ATP. All right? To make ATP, okay, or to make anything, you need to have the right supplies. You want to bake a cake, you got to have ingredients to make the cake. All right? To make ATP, we need ingredients. All right? One of those ingredients is phosphates. This molecule, creatine phosphate, gives up its phosphate so we can recycle and make ATP from ADP. We'll get into more detail about that, okay? All right, myoglobin, right? That is the cell's version of hemoglobin. It's going to bind oxygen, and then when you are exercising, or not necessarily exercising, but when that muscle is actually undergoing contraction, all right, it's the myoglobin that is going to provide the oxygen for the cellular respiration, all right? So we create that aerobic cellular respiration with oxygen so we can get as much ATP as possible. All right, what are the primary components of thick and thin filaments? Thick filaments are myosin. Thin filaments are actin, but we have the regulatory proteins of tropomyosin and troponin that are directly associated or on top of the actin. Okay, they're going to regulate this process. Oh yeah, this is perfect. This is a perfect place to stop. Okay, let's take a break. I know your brains are probably flipping out right now. All right, so we're going to stop recording here. Let's take a